Hello everyone, I am Microbiologist studying Dynamic Microbiology at Vivekananda Arts and Science College of Women, Sangagiri. This presentation was prepared under the guidance of Dr. R. Dinesh Kumar Sir, Assistant Professor, Department of Microbiology. Subject Soil and Environmental Microbiology. My topic is about space microbiology, microbial research in the space environment. Let's see about content including the space microbiology. Introduction to space microbiology, importance of studying microbes in space, the space environment, challenges for microbes, microbial behavior in microgravity, microbial survival and adaptation, microbes aboard the International Space Station, key experiments in space microbiology, application of microbial research in space, risk and challenges, the future of microbial research in space, recent discoveries and summary. Let's will see about the small video of space microbiology. The outer space is not friendly to life. The extreme temperature, low pressure, radiation can quickly degrade the cell membranes and destroy the DNA. This life form that show how fine cells in the Georgian die. The useless they bind together as a small communities. New research shows some bacteria can withstand the harsh environment. Today, we'll discuss about the space microbiology. Space microbiology is the study of microorganisms in the outer space environment. How they interact with the space environment is also called astromicrobiology or extromicrobiology. Mixture idea from microbiology, study of microbes, and astrobiology, study of life in the universe. Main goals to understand the origin of life, to search for extraterminal life. They began the earth. To know how microbes affect the human space exploration. Next, we'll see about the importance of microbes in space. Importance of microbes in space is mostly impact on astronauts' health. In space, microbes can become stronger and resistant. Astronauts' immune system become weak and microgravity. This makes astronauts as more vulnerable to infection. Then, biofilm formation. Microbes stick together and form the layer on spacecraft surface. This biofilms can lock have water filters as damage the equipment and make cleaning harder. This creates the problem in spacecraft hygienic and maintenance. Then, spacecraft contamination control. Microbes can spread inside the spacecraft and interfere with the scientific experiments. This closed evidence they multiple in quickly. Controlling them is very impact for long space mission on building bases on moon or Mars. Then life support and biodegradative system. Move on to the life support and biogenerative system. Life microbes are useful in space. They help recycle air, water, and waste. They can be used to grow food, make nutrition, and remove the toxins. Is very important. Long space missions like Mars travel, planetary production, it's prevent contaminating other plants. Example, moss within earth microbes is also avoiding bringing back harmful microbes from space, which ensure safety and accurate specific research. Next, move on to the challenges of microbes in the space. Microgravity, radiation, vacuum, temperature extremes, and closed system. First, microgravity affects cell process and nutrient transport. Then, radiation, a strong cosmic rays on damage the DNA. Vacuum, extremely cold and hot condition. Temperature then temperature extremes, very rapid changes, and closed system is waste product and gas accumulate. Then, microbial behavior in microgravity. Some microbes, like salmonellas, grow faster. Genus and stress response changes as more biofilm formation and spacecraft surfaces. They can become more resistant to antibiotics. Microbial survival and adaptation. Microbes survive in space by DNA repair, metabolic shifts, stress response, spore formation, genetic mutation. Let's explain the DNA repair. Fixing the radiation damage. Metabolic shifts, challenging how they use nutrition. Stress response, turning on special genes to handle the hot and pressure, etc. Spore formation as making the strong spores to resist the harsh condition. Genetic mutation. Some microbes mutate the faster. Example, Rhinococcus retinorans survive the radiation. Then microbes on ISS International Space Station. The ISS means International Space Station. Let's we see about microbe on ISSI. ISSI means International Space Station. Common microbes are Streptococcus, Pseudomonas, Bacillus, and Aspergillus. Then cleanness maintained by regular hair surface sampling, HPPA filters, humidity control, UV treatment. The astronauts' body microbes are also monitored. Key experiment in space microbiology, Microbial Observatory, Expo in Bionutrient Project and Pharmacet and Biosentinel. Then we explain the Microbial Observatory, NASA, Microbial Observatory, NASA, studies long-term microbes on ISS. As remind, ISS means International Space Station. Then X4 ESA as tested microbes as survival in the space export. Bionutrition product engineers microbes to make the vitamins and astronauts. Pharmacet and Biosentinel study drug effect and DNA damage in the space. Application of microbes in space, life support, bioremediation, and biomanufacturing, terraforming research. Life support mean microbes can recycle waste and CO2 and help to produce the oxygen and food. Bioremediation, they clean up the contaminations inside the spacecraft. Biomanufacturing, microbes can make useful things like bioplastic and medicines. Terraforming research as studying the microbes that survive the extreme conditions as help us prepare for the life on Mars or the moon. Let's we see about risk factor, pathogen risk factors. Then biofilm formation, contaminations and control, and mutation rates. First will pathogen risk. Astronauts as weak immunity can make the infection of pores. Biofilm formation. Microbes can form the layer that block the filters and damage the equipment. Contamination controls. How to keep spacecraft sterile in the closed environment. Future of microbial research in space. Mars missions, synthetic biology, microbial fuel cells, astrobiology. Mars missions mean need stable, healthy microbes for long journeys. Synthetic biology means scientists can design the special microbes for space takes. Microbial fuel cell. It means microbe can provide the energy and act as biosensors. Astrobiology help in research for extraterrestrial life. In the recent list of space microbiology and NASA clean room, that means super clean labs where bioscrapped or built. Scientists found 26 new labs for bacteria that were never known before. This bacteria can called extremophils, meaning this bacteria is called extremophils, mean they can survive in very throw condition. Their G help them live in the spaces with high radiation and big temperature changes and strong cleaning chemicals. Space is the place for women in microbiology. Thank you for all.